Welcome all of you. My name is Dr. Muhammad Fiaz from University of Education, Digital Campus. And today we will study about the most notorious insect pest of cotton, pink bollworm, whose technical and scientific name is Pectinophora gossypiella, and the family is Gallicae, and the order is Lepidoptera. And in this video, as usual, we will study about introduction, life cycle, damage, and control of the pink bollworm. Pink bollworm adults are small, dark brown moths measuring about 12 to 20 millimeter across the wings. The head is reddish brown in color with pale scales. Antennae are brown and the basal segment bears a pectin of 5 or 6 long, stiff hair-like scales. Antennae are brown in color and the proboscis is scaled. Forewings are elongated, oval, pointed at the tip and bearing a wide fringe. The ground color of the forewings is brown and they have fine dark scales that form vague patches in the region of the medial cells and at the wing base. The apical portion of the wing is dark brown with a transverse light colored band. Sometimes the wing bears a round medial spot. The hind wings are broader than the forewings. Pink bollworm is distributed throughout tropical America, Africa, Asia, Australia, including subtropical regions like Pakistan, Egypt, and Mexico. Interestingly, it has been eradicated from all cotton producing areas of the continental United States. The distribution map includes record based on the specimens of pink bollworm from the collection in the Natural History of Museum, London. Pink bollworm is an oligophagous pest. In a survey of okra, Dagenham, and Roselle in Egypt in autumn 1988, pink bollworm was found to prefer okra over cotton towards the end of the season when the cotton ball surface was hard. It is considered that pink bollworm in Pakistan is monophagous insect as it is only found to be attacking on the cotton only. Pink bollworm eggs are deposited on or near the cotton balls at the time of flowering. Young larvae emerge after 3 to 5 days in optimum conditions, entering the cotton balls shortly after emergence, where they feed internally within the pod, making a small hole to the exterior to allow air to penetrate. Larvae usually pass through 4 instars. After this, they emerge from the top of the ball, in contrast to the behavior of other cotton borers like Aries vitella and Aries insulana, which make exit holes in the lower half of the ball. Pupation takes place in the ground about 50 millimeter below the surface and adults emerge after about 9 days. Adults are nocturnal and the females begin laying eggs a day or two after emergence. typically laying 200 to 400 eggs each. Under optimal conditions, the entire life cycle is completed in 25 to 31 days. Under cool, dry conditions, pink bollworm larvae may undergo diapause in small cocoon in partially open balls in cotton lint, which is stored seed in the soil. Diapausing larvae normally emerge when conditions are more favorable, which are about late March or to early April, but may remain quiescent for up to 2.5 years. Extensive research has been undertaken into the use of degree day summation to predict pink bollworm development. Eggs require temperatures between 10 to 37 or 38 degrees centigrade to hatch. The developmental rate of all stages increases with temperature up to 38 degrees centigrade where mortality rises. Pink bollworm damage squares and balls. The damage to balls being the most serious. Larvae burrow into the balls to the lint through the lint to feed on the seeds. As the larva burrows within a ball, lint is cut and stained, resulting in severe quality loss. Under dry conditions, yield and quality losses are directly related to the percentage of balls infested and the number of larvae per ball. With high humidity, it only takes one or two larvae to destroy an entire ball because damaged balls are vulnerable to infection by ball rot fungi. The caterpillars feed on flower buds, flowers, and bore into the balls. When they bore into flower buds, they feed on developing an other anther and style and occasionally on ovary too. When they are found in flowers, the flowers do not open and give rosette appearance. The young balls, when attacked, are shed after a few days, but the larger balls remain on the plant. Locules are damaged and interlocular burrowing can be noticed. Seeds are destroyed and lints gets stained. The aperture through which they make their entry into the ball is closed, and it becomes difficult to differentiate between a healthy and infested ball. Let's talk about different control strategies of pink bollworm. The first one is biological control. Pink bollworm has been a target for biological control, particularly in Pakistan. Biological control agents were first obtained from localities in the belief that it was the origin of the pest. However, the matter is not settled, uh, and it may have come from the Indo Pakistan region. More recently, nematodes have been used as control agents in the United States. Pink bollworm is primarily managed with the host free period. Sampling balls is the most reliable way to monitor pink bollworm populations. The use of gossip lure, which is a sex attractant that disrupts mating when distributed throughout the field. It can be effective against pink bollworm when it is supplemented with cultural control practices that minimize the number of overwintering bollworms. The use of BT cotton will help prevent damage by pink bollworm. A recently developed transgenic cotton, Bolgar 2, offers suppression of cotton bollworm along with beet armworms, pink bollworm, and tobacco budworm. Eliminate the food supply for pink bollworm by cutting off irrigation early enough to stop the production of green balls by early September. Regardless of when the crop is terminated, immediately shred the cotton plants following harvest. Shredding destroys some larvae directly and promotes rapid drying of unharvested balls. If fall temperatures are high during September and much of October, leave crop debris on the soil surface for 2 or more weeks after the shredding operation to further destroy the larva. Be sure to comply with plow down requirements and cross disc and plow to a depth of at least 6 inches. In spring, irrigations can also be used to promote early spring emergence of pink bollworm. If cotton is being followed with cotton, pre-irrigate in February and plant as early as possible, following guidelines to ensure adequate soil temperature for germination and emergence. Plan irrigations of the crops to prevent even slight moisture stress and to promote maximum emergence of moths in advance of susceptible squares. Combination of biological and chemical controls have also proved successful. Application of trichogramma in combination with the chemical insecticides gave good control of pink bollworm in India and Pakistan. And Bacillus thuringiensis, which is also called BT, has been found to be effective in combination with chemical insecticides in Egypt. The pink bollworm, the project used the release of sterile insects, cultural controls, intensive monitoring with pheromone baited crops for adult males, and ball sampling. Pheromone application for mating disruption is very limited use of pesticides and the widespread use of genetically engineered cotton. Insecticidal control is hindered by the larvae being internal feeders. Moreover, resistance to insecticides develops, making it often more expensive than other methods. Chlorpyrifos, teflobenzuron, spinosad, and organic insecticides with high contact toxicity like carbaryl are extensively used to control pink bollworm. However, triazophrase at the rate of 1.5 to 2 liter and cypermethrin at the rate of 500 to 700 ml and triazophrase at the rate of 2 liters and endosulfan at the rate of 2 liters uh, can be prescribed to use to control against pink bollworm. So this was all about pink bollworm. I hope to see you in the next one. Until then, take care.